Hi, good morning, good afternoon. We're still waiting for a few people to connect, um, but we are very happy to welcome you all again. This is the 23rd lecture of the logical reasoning in human genetics. So we're going towards our 30th lecture, which is improving and increasing the size of our library of very important talks on the topic, the broader topic of human genetics. Today we are going towards an exploration of rare diseases in Africa with our first guest speaker from Central Africa, more exactly from the Democratic Republic of the Congo. Today's talk is, as usual, a partnership between Columbia Global Centers Tunis and Institut Pasteur de Tunis. And also we have with us as a partner the Columbia Global Centers Nairobi, which covers the region of East, Central, and Southern Africa for the Columbia Global Initiative. And they are with us as a partner for the second time. As I mentioned, we have the honor to welcome Professor Aimé Lumaka, Professor of Genetics and Pediatrics at Université de Kinshasa at the DRC. He's also the chair of the African Rare Diseases Initiative, and he will guide us through essential insights and initiatives aimed at advancing genomic medicine through rare diseases research in Africa. This talk will be moderated by Professor Ghada El-Qamah, who is Professor of Clinical Genetics and who is joining us from Cairo, from the National Research Center in Egypt. Ghada is also a member of the advisory or steering committee of the Logical Reasoning in Human Genetics. Her research is mainly around the study of medical genetics with a focus on inherited rare disorders. Ghada, the floor is yours, and you will moderate the discussion with Professor Lumaka. Thank you.
Thank you, Youssef, very much. It's a pleasure to welcome you, Prof. Lumaka, or Aimé, if I may make it smoother.
Sure.
As you heard, it's all about, it's very innovative. We are very concerned about logical reasoning in genetics, not just taking info as they are. And as we are in the rare disease disorders day, it's a very good chance to seize and discuss the problems in an African context with you present. And African population are really large and very diverse. And I would imagine we're having lots and lots of rare disorders among this population with variability. I haven't studied. I don't think it's a continent, of course, but the degree of diversity all the world knows about. So it's great to know about how you started the network. What did you find within this network? How can we benefit from this research? How to include us all in this? And if you can add the logical thinking concerning your studies all through this long journey. Concerned with the rare disorders. Please, the floor is yours, Aimé.
Thank you very much, Ghada. Thank you for the invitation, and thank you for the assistance I've received since I received my first invitation until today. And I am going to share my screen. Let me know if you can see my screen.
Yes, yes, we can.
Okay, so today I have the pleasure to share with you the African Rare Disease Initiative, an initiative that is willing to advance genomic medicine through research focusing on rare diseases in Africa. I am from the University of Kinshasa, as mentioned in my introduction. Because it's a logical reasoning or thinking series, I try to focus my presentation around the logic behind this whole initiative. As we all know, rare diseases, we in Africa, we prefer the European definition, that is any disease that affects less than 1 in 2,000 people is considered as a rare disease. And second, for people living in the USA, a disease is considered rare when it's affecting less than 200,000 people in the USA. Furthermore, although each disease is rare, but if we take it together, about up to 6% of the world population would be affected by rare diseases. The NIH actually counted up to 7,000 rare diseases. And all those diseases would represent 25 to 30 million affected people in America. To put it into perspective, that would represent 1 in 10 American, or 1 person in each elevator in the USA, or 4 people in each bus running into the USA. In Africa, that would be 42 to about 71 million people affected by rare diseases. This is an enormous amount of people. The number is so big that would represent even multiple countries of people willing or seeking for diagnosis or care for their diseases that are receiving little interest thus far. Up to 72% of those diseases appear, they manifest, they have the onset during the pediatric window of age. And up to 70% of these diseases appear in pediatrics, and 72 have genetic underlying pathophysiologic mechanism. If we look at the most known diseases, genetic diseases, we learn that those diseases have different distributions across the globe. For instance, we have diseases like cystic fibrosis or phenylketonuria that affect more people of European descent as compared to the sickle cell anemia, which affects more people in Africa or in Indian region, all those intertropical regions. And if we are looking at the entire rare diseases problem, what we would see is that the data on rare diseases in Africa is similar to this picture from NASA, where you have data from all the places that you see here have lights. And when it comes to Africa, and particularly this Central African region, it's a dark hole. We don't know exactly what is going on there, which diseases are prevalent in this region, what is the genetic pathophysiology behind those diseases, how we can prevent those diseases. If we would like to advance genomic medicine, we would like to need to do make discovery and do the implementation. And both the discovery and the implementation are data-driven, which means that we need to produce data. We need to extract information from those data. We need to compare these data to generate knowledge, and then we would be able to translate this knowledge into action so that we can improve not only our knowledge, but we can also really use this knowledge to change the health condition of our patients and our families. The more we have data, the more we can make discovery, but the more we have data that are of good quality, the most quickly we can make those discovery. Data are needed in the entire genomic medicine process, starting from the patient data and feeding those data to the lab, and the lab feeding the data generated by the sequencing into different tools and platforms so that we can get really the sense of which particular variant in the genomic data actually matches the phenotype of the patients after being filtered against all the information that are available in the public resources. For the moment, the phenotype of some of the known diseases are not well described in African population, and data in the databases that are essential for the interpretation of genomic data are really poorly represented in terms of African data. If we would like to increase the volume and the quality of data, we need to start by where those clinical data are mostly found, which means the health record of patients. We see that in the All of Us project, for instance, in USA, we see that in the UK Biobank, how the medical record of patients are actually driving the discovery and are pushing forward research. In Africa, in many countries, especially in Central Africa, medical records remain handwritten. So there's a need to digitalize those health records to facilitate the aggregation and to make it possible to search the data and to find the relevant information in the data. We also need to recognize that even in USA or in Europe, any patient affected with a rare disease, if he's lucky, might have a chance during the lifespan to meet one geneticist. But most of the patients, for the most of the part of their life, would be seeing the GP, they'll be seeing the neuropediatrician, the cardiopediatrician, the neurologist. Those are people that are actually in daily basis following patients with rare diseases. If you are working in a context or where your electronic medical record is handwritten, it means that you have multiple people actually producing handwritten notes. And the more you have people, the more you have differences in the way they are actually writing the report. They are actually generating multiple reports in multiple languages. That is a problem. If we are willing all to advance genomic medicine, we need to harmonize the way we are collecting information, harmonize the tools we are using to collect the information. So we need to capacitate those people that are non-geneticists in how we collect data, the use of HPO terms, for instance, how we describe the phenotype of patients, and how we approach patients when it comes to counseling, explaining genetic tests, to ensure that they are taking note in the appropriate way and they are communicating with patients in the way that would be the most univocal and the most appropriate, as we would like that to be. So we responded to this RFA from the NHGRI with the initiative that we call the African Rare Diseases Initiative. First, when we started this about 3 to 4 years ago, the goal was to have a network of 14 African countries involved in one network in rare diseases, all working in 3 different fields, which is conducting research on African patients with rare diseases, and also developing the capacity to do this, the bioinformatic capacity, and also organizing training sessions for people from those 14 countries. Unfortunately, the application didn't go through, and we were advised to reduce the size of the project at the time. So we went back with the initiative, but at a single country level as the proof of concept of the ideas we were putting forward. We were lucky this time. The application was funded for an R1 that started last year. So what is it about at the moment? The first aim of the African Rare Disease Initiative is to develop rare diseases infrastructure for rare genomic research, rare disease genomic research in Africa. When we are talking about infrastructure, it's not about buildings. It's about having a network of people who can work together from different geographical fields from different level of training. We have mostly pediatricians, but we also have people that are GP, but they are actually in charge of pediatric services in the regions where you have no pediatrician at all. And when the network is there, members of the network would actually sit together and discuss what should be the minimum content of a health record. Once the minimum package has been decided, this package can now be transformed into an electronic health record that everybody within the network will be using to collect data from patients. The capacity or the infrastructure building also includes the development of a mobile app that would be able to explain to patients in the languages. The Democratic Republic of Congo has about 220 different dialects, but we have 4 major languages. These are the 4 languages that we are actually targeting for the mobile app, so that wherever someone is in the country, you'll be able to understand what we are explaining on the mobile app, and would be able, most importantly, to connect to the network, identify the most closely located sites of the network. We are also developing a website to provide information and also to keep people updated on the progress of the network. The second aim of the African Rare Disease Initiative is to actually capacitate African researchers in rare diseases, and especially in the genomic aspect of the rare diseases. That includes training people, in-person training, but also online training through different training media. 3 areas of training has been identified and are the focus of the African Rare Disease Initiative. That include training people with the bioinformatics skills to be able to analyze genomic data that are produced in the African Rare Disease Initiative and expand this expertise, share this with other people in the country or in Africa. The laboratory aspect is also important for us, because we would like people from remote areas in the country to be able to do the minimum that is required. That is extract the DNA. I have one slide, that picture that I would show you. If you would like to travel between 2 corners of the country, sometimes you need to go to other countries, neighboring countries, or you need to go to Ethiopia or to go to Kenya so that you can take your connecting flight to come back to the country in a different places. It's really difficult. You cannot shuttle DNA or blood sample in these conditions. People need to be trained in DNA extraction. And then we also have the clinical training, train people on how to conduct consultation with the patient with rare diseases, depending on the clinical handle, the major symptom of the patient, and also how to conduct counseling. For the training, we are actually relying on existing material from ClinGen, the American College of Medical Genetics, the environment in the UDNI, and of course the DSI Africa, that is a consortium on data science. The third aim of the African Rare Disease Initiative is actually to prove how this network can efficiently advance genomic medicine. We have decided of inclusion criteria because we would like to recruit 300 probants in trios and offer them whole genome sequencing. And those patients would be recruited along those 4 categories, clinical categories: patient with developmental disorders and or intellectual disability, epilepsy that should be familial or syndromic, and myopathies and neurodegenerative syndromes, as long as they appear early in life. And we are also interested in congenital malformations, but not isolated malformations. Given that we are in a country or in a region where you have a lot of acquired causes that may cause phenotype that mimic those we are interested in, we have decided to exclude patients who have history that can suggest an acquired cause that include trauma, especially brain trauma, severe infections, especially meningitis, perinatal asphyxia or suffering, toxic substances, and the extreme prematurity. We are especially interested in some groups that are less explored for the moment in our country. That include the indigenous population, the groups with high consanguinity or endogamies, and group with an unusual aggregation of particular phenotype as the informations are available thus far. For the recruitment process, we have established a flow that starts with patient contacting the network through the mobile app or through the website. Once a family has contacted the network, they are actually reoriented to the closest site where they can go for the clinical consultation. The clinical sites conduct consultation on a monthly basis, at least one session of consultation for each of the sites. What is interesting here is that each of those consultation sessions are actually a hybrid session. You have the physician from the clinical site receiving patient, offering consultation, but you also have the hub and all the guests from outside that we are inviting depending on the expertise and the information the patient provided when seeking contact through the mobile app. So we are actually attending the consultation virtually, and the physician on site is conducting the consultation. We all ask questions and we guide the clinician on site. It's a training opportunity for them to learn in practice how we do things in the genetic field. Once a month, we meet together to discuss the patient that we have seen in consultations. And then we, after this review, we decide which patient actually matches our recruitment criteria and should receive whole genome sequencing. Patient information are entered into the cipher. The sequencing is going to be performed at the Baylor College of Medicine. We actually opted for the same quality as the All of Us project. So data generated through the African Rare Disease Initiative would have the same quality standard as the All of Us data. And the analysis is going to be conducted in the cloud environments. And once a month from the moment that data would start being available, we will have a variant review meeting to discuss variants that are actually solving the case and also variants that would require reverse phenotyping to reach a final decision. We are developing a registry of patients that is linked to the electronic health record where we would actually fed the data. And we have a large plan for data sharing and data dissemination that include publication and all other type of sharing. The entire flow is really complex given people and tools that are involved. The recruitment involve the sites, involve the tools that we are using to get the match based on the face of the patient or to store the information in the cipher. And then we have the sequencing that is going to be performed at Baylor, and data will be transferred. The raw data for the sequencing machine will be transferred back to us for the entire analysis to be performed by the Congolese team. This is important for us to really retain this bioinformatic analysis of data and the downstream clinical interpretation, because these are training processes and it's important for us to train people during the data analysis process. The dissemination is broad, include the deposit of clinical pictures in the Atlas of Human Phenotype Ontology, publication in PubMed, deposit of variant in ClinVar, matchmaking, and also making information or variant available through the cipher. These are our partners for the training, of course, the NIH and our scientific advisory board. So where are we now since we have started? As I mentioned, the project was officially funded in June, and we started the 1st of July. Until now, 7 months or 8 months into the project, we have successfully developed the network into the country. This is the country. The country is 1/4 of the USA in terms of territory. So it's a big country. And we have sites across the country. We have the hub at the University of Kinshasa. We have university hospital or provincial reference hospitals involved into the network. These are the names of the hospitals. So we have now been able to sit together and start or finalize the minimum content of the medical record. Last week, we had a big meeting where all the representatives from all the sites, the clinicians from the site and the lab technicians from all the sites came actually in our office at the University of Kinshasa. We have those meetings for 1 week. Where we, among other things, we actually agreed on the minimal content of the medical record that our informatics team is now going to implement in the electronic health record. The website is also running. You see the addresses here. It's the initial version. We are now working to populate this with more information and also produce an English version. After the website is up now, the developer team is starting this week with the mobile app. For the second aim, this is the picture that I mentioned before. These are the conditions that people are actually facing on the ground in their respective regions to move from one part of the country to another. And the delegates have to face these tough conditions where they were traveling for 1 day or 2 days along a distance that someone in USA would be traveling in 2 or 3 hours. They needed 1 or 2 days to travel those distances. When they were in Kinshasa, we actually organized courses for them, the theoretical courses, but they also went to the lab to actually see what it looks like, what we do when we have blood samples collected. We also had practices for clinical consultations and counseling. Our PhD students and master students are actually participating in the DSI Africa. And we were lucky that our collaboration with the ACMG has actually allowed us to get from the ACMG the education material for the 100 syndrome that everyone training in genetics should know. So we have those documents now. In terms of the practice, the investigation of the genetic causes of rare diseases in DRC, we have started with the virtual consultation, those hybrid consultation. For instance, here you have Dr. Germain Mudumbi from Bukavu with 2 different families. We sat through Zoom, we talked to parents, we explained, and we recruited those 2 patients. These are the sites that have already recruited patients where we have already organized consultations. And this activity is now set to speed up in terms of pace now that everybody has been trained in Kinshasa and they are returning back with fresh knowledge. And during the meeting that we have had last week in Kinshasa, we actually went through the situation of rare diseases in each of those provinces. We were able to identify 2 groups of high interest. That is the indigenous groups, where they are located, which are the most observed problems in those communities that would require the involvement of the African Rare Disease Initiative to provide diagnosis or to provide a better understanding of the health situation in those communities. We have also identified a community that is geographically isolated because they are on the lake. It's an island on this lake that is, it's somewhere here that is separating the DRC and Rwanda. So now we are going to continue investigating, dive deeper into those groups and see how we can provide our help to those communities. Next week, I'm here in the USA today for a meeting, and next week I'm traveling down to Texas so that we can officially sign the MTA. When I'm back in Congo, we can start sending samples for sequencing at the Baylor College of Medicine. So this is where we are. This is a big effort that is made by multiple people that I wanted to acknowledge through this slide. And I also thank you for invitation once more and for your attention. Thank you very much.
Thank you so much, Aimé. It was so interesting to hear about your presentation, Yani, and your initiatives. It's so sad we did not join you yet, but you know we are very ready to join. I know. No, it's really, I'm very glad about. I remember the first time we met, you were so enthusiastic about keeping the African samples in Africa and implementing African research in Africa. He was much younger than now, and he was shouting with his high voice.
Yeah.
We're not going to give you any more samples. We're going to work in Africa. And after all those years, I'm very happy to see that you're an example of a dream come true. And it's not only that, you're giving a very good example of what I am usually discussing with my colleagues and students about the brain drain. I would think of you as one of those brain drains stolen from Kinshasa to Belgium. But actually, yes, you came back with a very good project and you're helping your country, and not only your country, you're helping the huge number you mentioned, a huge number, you said 40 million African people suffering rare disorder. That's almost a large populated country. It's It's a scary number, yes. I don't want to take it all, I can discuss disorder with you for hours, but I cannot see any questions in the Q&A now. So I would start by asking my colleagues, the panelists, do you have any questions?
Well, am I frozen? Yeah, okay, there I am. Yeah, I think it's great to be doing this sort of stuff on the ground there. The first thing, of course, that comes to mind as a geneticist is about the ethnic diversity, because there's quite a lot of that, right, in DR Congo. And so how do you deal with the fact that you have so many different ethnic groups that you deal with? Presumably rare diseases are going to vary between those groups more than they do elsewhere, because that's the nature of genetics, right? You get the, population specific things. I've worked most of my life in unusual places, like, where small populations where they have unique sort of variants in the region. So presumably the picture of these diseases and their nature and frequencies is quite different than in other countries in between groups. Is that true? Or can you say something about that issue?
Yeah, thank you, Joseph. We know that because of the diversity, the type of diseases and the variant causing those diseases may vary across the groups. But because we are actually lacking data, we don't have data, for instance, in DRC to support that. We have patients that we have molecularly diagnosed at the moment, but because of the rarity of those diseases, these are just the ones. The first, and if you are lucky, you have 2 or 3 in the country. It's not yet sufficient to draw any interesting conclusion yet. But what I can say is that we have been involved in the facial analysis of patients. And we have done that comparing the faces of individuals based on the geographical origin. I cannot say that it really matches the genetic background, but we took that as a proxy for the background. What we saw is that there are differences in the facial morphology of people within the country. And we did that also with our neighboring countries. Actually, it's a funny story. We couldn't publish that because of the sensitive nature of those studies. We actually tested people from DRC and from the neighboring countries. We compared them to people from Belgium. And when I'm saying people, is Down syndrome patients from DRC, from the neighboring country, from Belgium, and we compared them to Down syndrome patients from Guadeloupe. It was really interesting to see that patients from Guadeloupe were actually fitting between the DRC patient and patient from the neighboring country. And the area under the curve was really one, nothing to do between the face of Congolese patient and the face of the neighboring country, where we were sharing similarities with patients from Guadeloupe. It's historically understandable because those people came from the eastern part of Africa. If we trace back the slavery trade, they were coming from the western part. That's why we had that similarity, some overlapping features with DRC patient, but nothing to do with patient from the neighboring country on the eastern part. That means that we need to continue this work, really try to see at the genetic level what that means. And we are lucky to have the network stretched out across the country, and we really hope that we can put genetic data on top of those phenotypic data that we are observing.
Yeah, no, I think it's really interesting. I know that I'm sure my colleague Joe Lee, who showed up, he works on Down syndrome, so I'm sure he's going to have questions for you about that. But I was wondering, just thinking off the top of my head, that things like, for example, phenylketonuria, there seemed to be almost nothing reported in Africa. And yet it's one of the most diverse genes. It's a huge gene. And in Africa, there's all kinds of haplotype diversity. Is there any, is it just that it's under ascertained and people don't notice it? Or is there really nothing there? And I was also wondering if that has anything to do, for example, with the way people choose mates or something. Is that any different than in, say, like in North Africa, there's a lot of consanguinity. So you get a lot of rare diseases because of that. But maybe, is there something different in the way that works in sub-Saharan Africa?
Yeah, definitely something to be explored that we are ignoring at the moment.
Well, it's fascinating. So I look forward to seeing your research over the next decades.
Thank you.
Yes. Do you have any knowledge about the consanguinity level in Central Africa, I mean?
Yeah, so during the discussions that we had last week, we really wanted to have the insight from people from different parts of the country. We didn't really identify consanguineous communities per se, but what we have is people that are culturally isolated by the culture.
Yes.
They always want to marry people from certain background due to historical reasons. We were able to identify those groups, but we were also able to actually note the evolution of the cultures as the religions is influencing the culture, as the education is influencing the culture. There have been adaptations in the country. Let's say that culture, for instance, that where you have people marrying first cousins. Now the culture has shifted. If you would like to marry, if a lady would like to marry someone outside of the tradition, this incoming husband has to pay a price. It's in terms of money. You pay the price and then you marry. So that allows people to escape this strict cultural rules or limitations, people are now able to marry outside as long as this incoming husband would be able to pay the price. We don't have consanguinity. A lot of them per se, it's more these endogamic communities, and we have isolate. People have not really been able to cross natural barriers, the forest, the rivers. People have not really been able to evolve beyond these natural barriers.
So those natural barriers are part of why Africa is so diverse then, because it prevents sort of admixture between groups. Is that what you're saying?
Yes. Yes, this is what we also took out of the discussions we had with our colleagues last week.
Oh, fascinating. I look forward to seeing more of the molecular data over time.
Yes.
I was thinking that these natural barriers would create a much more endogamous population because you're stuck together. It's like how it is in Nubia, in Maasai tribes. I was surprised. You have lots of Maasai who are still living as tribes, keeping their tradition in their isolated locations and things like that. I don't want to take more time. Yusra has been raising her hand for some time. Please, Yusra.
Sorry for thank you very much. No problem. I'm so sorry because I'm driving, so maybe the voice is not clear. Thank you very much, Aimé, for the excellent presentation. My question is about comorbidity, and if the rare diseases, the frequency of the rare diseases in Africa, if it's different, this concept, is it affecting the frequency of other disease or the comorbidity within the same families?
Yeah, very interesting. I would say that the comorbidity is one of the arguments that we are using to convince our colleagues to refer patients to us. There's this study that I gotta mention my affiliation with the University Hospital of Liège, which I'm not anymore. I initiated a rapid whole genome sequencing study in the neonatology setting. We identify 55% of newborns with a genetic condition. In DRC, one of the first patients that we actually diagnosed was a patient with Williams syndrome. It's a recognizable phenotype, but this patient has been followed by multiple pediatricians and neuropediatricians because of a history of meningitis around 2 years. And for them, all the remaining symptoms were the sequels of this meningitis. We have people that are followed or they are presenting a true infectious diseases, but they also have a genetic condition that has nothing to do with this infectious disease. Or they also have infectious disease that may be explained by the vulnerability that is created by this genetic rare diseases. We have people with G6PD deficiency. We see a lot of this is coming out as the most common incidental finding in our genomic results. They have something else, but they have G6PD, and then they will go to the hospital when they have malaria. They would be actually followed by our colleague from the pediatric nephrology because they have acute kidney injury. So we see that a lot. That's why we never really say true. Definitely, this is an infectious disease. This is not a communicable disease because the frontier is really, really narrow between both in our setting at the moment, given the level of medicine we are able to practice and the existence of the lack of some of the investigation for biomarkers that is important.
So one thing that, no, sorry.
No, sorry, please.
I was just going to say, it seems to me that, well, first of all, there's genetic components to infectious disease, of course, right? You know, in terms of resistance and whatever. But I was also just thinking a lot about when you define rare disease, and if it's based on frequency, what's the denominator? In other words, is that frequency in Congo, in Africa, or in an individual ethnic group? Because we see that all the time, right? In Finland, for example, you have diseases that are relatively common that are absent in Sweden. And in Sweden, you have diseases that are absent in Finland, but they're actually quite similar compared to some of your ethnic groups. So do you need to define it per population or per ethnicity, or is it nationwide that you're using? In which case I would understand why you would lower the threshold.
Oh, yeah. There is a need to have appropriate thresholds. We see multiple countries have been able to develop their own threshold. For the moment, because we don't have the data, we are just using what is available in the publication. Otherwise, your manuscript will not be accepted. So this is where we are at the moment, but the need is there. We need to redefine. Let's take the example of sickle cell anemia. One in 3.5 in DRC is heterozygous carrier. About 2% of all the newborns are homozygous, which really puts this beyond the threshold for any definition we'll be using. The European definition of the USA definition, it's really above it, cannot be considered. But if you look at on the other side, the fact that it still receives less attention, it requires research, it requires investment. If you put this out of the agenda, it becomes a loss of forgotten disease. So there's a balance between just pure statistical definition and the operational definition. I would put it on the list because it would drive attention on this disease. There's a balance to be found, country dependent. What we are doing now is trying to redefine, for instance, the newborn screening list. What would be, if we would like to develop a newborn screening program in DRC, what would be the diseases? We have a PhD student working on that. And we are trying to actually use the genomic data and also use patients that are in intensive care units, neonatology and pediatric intensive care units, actually to have an idea of which diseases are circulating in DRC that may be actionable based on the WHO criteria and would deserve to be put on a list of disease that deserve to be screened in DRC, because we feel that it's not possible to replicate something from another population or another country. We are trying to start with the newborn screening. Maybe we'll be able in the future to redefine really the rare diseases for our country, taking into account the actionability.
Yeah, I guess eventually you have to, because presumably there's different variants and different populations that are different from the ones they know about elsewhere, even if you can identify them. Yeah.
Mm.
And as an African network, I think it's important you might help other African countries to also redefine their priorities as neonatal screening programs. But you're only speaking about rare disorders, because in the field of genetics, it's very rare. It's very difficult to define what's rare. I would add to what to your opinion, Joe. Actually, when you're a geneticist, Aimé, you find lots of physicians coming seeking your help, like nephrologist, GIT, intractable diarrhea, and they want to know the genetic background. And lots of neonatologists are suffering this problem. They're facing this problem of neonates having intractable diarrhea, and they are, they don't know what to do. And they want our help as geneticists to try and define a genetics cause, which is very difficult. So how do you set priorities in your research? If you're thinking only of rare, it would be a bit difficult or unfair to those children not to include them in your neonatal screening. So maybe it would be good to redefine your search, decrease the trios.
I mean, on some level, what you're saying, it's almost been in my mind that rare isn't the issue, it's simple. right? If it's genetically simple or, closer to monogenic, where there's genetic factors that are big players, those are the ones that are important. Now, I mean, in Europe, they tend to be rare because we have big populations and because of the mating structure. Probably it's different in Congo because they haven't looked at these things enough yet. But I'm guessing that it means the more genetic diseases is what we're really focusing on, not just because they're rare, but because they might be simpler, right? In which case, you don't even need a justification for sickle cell because it is genetically fairly straightforward. Yeah. So it's almost like that's the criteria rather than frequency in some sense. Yeah, it's just a surrogate for simple. We hope we're simple.
I think Joseph nailed it. This is really the thinking that is ours. There's a big hospital in DRC, in Kinshasa. They have developed a network for the treatment of patients with diabetes in Kinshasa. It's really known and it's taught in the medical schools because it's a bad example, but it's the only one that we can follow and apply in this. What they are doing is that if we have one bottle of insulin, we have one vial of insulin, we can give this insulin to more people instead of giving to one person, and just give to the more people you can today and expect that tomorrow you can have another vial of insulin to give to people. What I'm trying to say is that when we have the opportunity to test patients, yes, because patients are not paying, or everything that we do is free, they are getting access to these tests for free. Because we have a limited number of sequences that we can offer to patients, we prioritize. We give more chance to the most likely genetics. We are actually looking for those phenotypes where the chance of identifying the genetic underlying cause it's really big. That's the idea behind the construct of our recruitment criteria. We have been in the DDD Africa project. We are now in the African Rare Diseases project. We have been in the Illumina IHOP collaboration. It's all the same thinking. Luckily now we have more chance to move to other projects. We were talking about agenda with Yoso before. What we did, as opposed to other countries, we said, okay, let's actually sequence people who have COVID. Let's get this opportunity to sequence people who have COVID and develop kidney injuries, and they have sustained kidney injury after COVID. Let's see why they have sustained kidney injury after COVID. And these are opportunities that we are having, that are allowing us to diversify the phenotype of people we are offering sequencing. Only two ways we are getting sequences: it's philanthropy, it's research, and we need to set criteria.
Well,
so I mean, do you have a expectation or any numbers on what percentage of truly unusual young pediatric disease patients you actually capture, or what's the plan going forward? In many Western countries, that percentage probably reaches very high and gets higher all the time because I guess doctors are aware that there are networks of people interested in this research. So, people are connected and information flows widely, but you're just starting out. I assume right now this number is going to be very low, but is there what's your expectation going forward?
You mean for pediatric genetic conditions?
Yes.
Okay, thank you. So if I may refer to data from the, because for the DDD Africa, we performed whole exome sequencing. We are now in the process of doing the analysis of data. And for the collaboration with Illumina, the IHOP whole genome sequencing was performed. If I look at the first batch, the first and the second batch that were already fully analyzed, and I would combine the likely pathogenic variant and the uncertain significance plus variants, because they are not known yet, not published, no functional analysis. Then we had to go back to do the reverse phenotyping, see the patient again, ask for radiograph to actually get more information on this. What we have not been able to do is to do systematically the extended segregation analysis within families. That is something that we are considering in the African Rare Disease Initiative. So what I was saying is that when we put all those strategies together for those first 2 batches, we were able to reach 45% of a resolution, which means that the recruitment criteria are actually the proxies that define our result yield. If we apply the same systematic testing as applied in Europe, where a patient would come to the pediatrician or the neuropediatrician and go out for the CGHRA and other tests, the yield would really significantly drop. It's just because we are drastically selecting them. That's why we have the 45%. And I expect this to drop as we are expanding the phenotype.
Oh. Well, I mean, 45%, I understand it's probably an overestimate given your selection criteria, but that's still impressive to me. And you know, what percentage of patients with these rare pediatric diseases do you actually think you're capturing in the country?
At the country level, I cannot really
I mean, I'm assuming you're just reaching the tip of the iceberg. It's just, you know, 1% maybe, but you have the mobile app, and I see it's wonderful that doctors can kind of link and reach out to you that way. But so presumably you also want or looking for some kind of advertising campaign to increase awareness. So ideally, you know, any doctor out there that sees a very unusual pediatric patient with some condition that they don't recognize, ideally would kind of somehow reach out to you in the network and gets connected.
Yeah, presumably there's also a lot of like infant mortality that's unreported due to these rare diseases as well, especially in countryside, man.
Yeah, definitely. And another element that we need to take into account is on top of this selection criteria that we are actually setting or defining, there's also a natural selection of those patients. Joseph mentioned the fact that some of them will die early, but it's the economic proxy is that because there's no reimbursement of health care in Congo, you need to be able to pay your transportation to come to the hospital. You need to be able to pay for the consultation. And we are doing a lot of our campaign, because we do campaign, we go a lot on the field. We are doing most of the campaign in specialized education schools. Just in number, one of the big schools in Kinshasa is charging $1,800 a year. $1,800 a year, which means that it's 18 times a salary of just the normal person in the country, 18 times the salary. You can only go or send your kid there when you have this type of means in the country. The other thing that we have noticed when we go to those schools, it's another element of selection. Parents only send the child where they have a hope that the child will recover. They don't send the most severely affected children in the country. So for the moment, this category of most severely affected individual, we don't capture them. We don't capture the information from families that have low income. So these are really categories that are escaping. We don't capture those.
It's a big challenge. You know, in the West or rich countries, we're discussing, at least conceptually, to sequence every newborn at some point, and it's just part of the regular healthcare system. Then presumably would get everybody hooked up to databases of appropriate type, and you're starting at the extreme other end. So it's a big task, but it's important. It's fascinating. So best of luck.
Thank you. Thank you.
I have a general question. Sorry, I missed the early part of your talk, so you may have covered it. But my question is this: you said something about you have over 700 rare diseases in your definition, and I was wondering.
Sorry, I missed a part.
You said there are over 700 rare diseases that you have cataloged. When you look at those data, how many of those rare diseases that are rare in your Congolese population are actually common in the Western population? Vice versa, how many are rare in the Western but very common in Democratic Republic Congo?
Okay, so I guess you are referring to the NIH count of 7,000 rare diseases. For the moment, as I mentioned, we are suffering of paucity of data. Very few data are available. And the diagnosis that we are actually encountering is more diseases than more patients for one single disease. It's like one first. That's the problem that we have at the moment. We cannot really say which disease is the most prevalent or significantly differing. But I've mentioned GCSPD deficiency. This is something that is really popping up in our analysis. You see this, it's like sickle cell anemia. We don't look for this, it just pop up out of during the analysis. GCSPD is also the same, it just pop up during the analysis. I hope that when we finish the analysis of the DDD Africa samples, 150 patients on top of the 200 that we have with Illumina, we would get the second, maybe the third, and start seeing the trend. But for the moment, it's unfortunately most of the first for different diagnoses.
Yeah, one more question. I do research on Down syndrome. So I'm just curious, when I'm looking at the literature, I usually say that African Americans and Hispanics have a higher prevalence of Down syndrome in the United States. The life expectancy of people with Down syndrome used to be about 10 years until more than 40 years ago. And then in the past 40 years, they have more than doubled their life expectancy, so that people easily live to 6 years of age. What is it like in Congo, Democratic People's Democratic Congo?
Okay, thank you. Down syndrome is, I always joke about that. When my colleagues in Belgium are unable to recognize an African patient Down syndrome, I always joke about that and say, my wife is able to recognize this. We recognize them. We don't need genetic training to recognize Down syndrome from a Congolese. Yeah, because my wife is not a physician, but she recognizes Down syndrome on the street in Congo. So we have, I would say Down syndrome is one of those frequent genetic conditions that we are seeing that are coming to our consultation. We don't really have statistics in the country because of the end note, as I mentioned. We have the end note, we cannot pull data together and generate statistics. What we have observed is that when they don't have a heart disease, they can live until the adult age. We see these are the Down syndrome I grew up with in my environment. I can still see them there in the city. When they don't have heart diseases, they tend to live longer and become adults in our community as well. The heart defect is really the determining factor for the life expectancy at the moment.
Yeah, more than half the people have heart defects. So in the U.S., most get have a surgery early on in life. So that probably extends their life considerably.
Oh.
Thank you.
Okay.
I noticed in your list, your map where you had all the hospitals you're working with, that you even had people over in the east near Rwanda where there's an ongoing conflict, right? I mean, are you able to work through that condition with the hospitals there without problems?
Yes. When we had the workshop last week, at some point we were convinced that our colleague from the eastern part would not come because they actually have to, people from Bukavu, they have to go through the lake until Goma and take the flight from Goma to Kinshasa. And the lake is really the border between Rwanda and Congo. We also have our colleague from the extreme north, and they couldn't travel coming to Kinshasa, but they had to go through Uganda and fly from Uganda to Kinshasa. Actually, we were able to have all of them in Kinshasa by using those approaches. But yeah, actually, it's possible to, they are still working. And in terms of scientific word, we still have very good collaboration with our colleagues from Rwanda. The only thing that we say when we meet, let's put the politics aside, and then we can work together and do amazing things together.
Yeah, that's great. Yeah, science always is good for building these kind of bridges, right? Because we just look at that science and don't care about the other stuff. But it's probably really interesting because the ethnic composition is quite different, right, in the East from in Kinshasa.
Yeah, the province of Kivu in itself is in a Thai country because you have since 1994, the genocide in Rwanda, you had an influx of people from Rwanda, and it has changed the composition of the eastern part of the country. It's a good element for genetic research, but it's also a problem, it's a sociological problem. You need to be careful because the genocide started because people
thought about
it different, and the difference started the war. So when you are dealing into those differences, you need to recognize this and be really careful on what type of research you can do, what you cannot do.
We had the same problem in Finland with the Sami population, which is an indigenous population in northern Finland. And they wanted us to do genetic tests to say who's a Sami and who's a Finn because they get different rights for land use and so forth. And we're just like, no, we're not getting involved in that. Genetics should be used for good things, not for creating more problems.
Yeah.
But I mean, in terms of a lot of these questions like Harold was asking about prevalences and the epidemiology, how about if you just look at Kinshasa, forget about the whole country and just look within the city where you have the best infrastructure, where you have the most ability to capture the population? Can you make more definitive statements about what the situation is there? If we, forget about the rural parts for now and the rest of the country and just think about in the one main area where you work.
Kinshasa is an interesting city because nobody really is from Kinshasa.
Sure, of course, like New York.
So nobody is from Kinshasa, and it's generating interesting lessons. We are learning a lot just by looking at the result we are getting from Kinshasa. We see, as expected, a lot of autosomal dominance, but we also have extreme rare diseases with homozygous variants, and the father and the mother has nothing in common. One is coming from the east, one is coming from the west part of the country. It's really challenging in terms of trying to find the explanation, trying to explain why it happens. And
Are they really homozygous or maybe compound heterozygotes or something? I mean, have you done the molecular analysis to see that? Or
Yes, we have a family with a primary mitochondrial defect, the MIST-01 gene, and the variant is homozygous. And they have actually 3 children that are clinically affected, and they are experiencing more and more severe phenotype. The father is from the central part and the mother is from the western part of the country. We still see this happening in the country. But as I say, we have more autosomal dominant diseases in the country. That probably it's something that is expected, as Brenner always say, you can expect that to be the first mechanism, and it diminishes the number or the likelihood of having autosomal recessive diseases because people are coming from very, very far away from each other. This is what we are seeing at the moment. But the fact also that we are working with whole genome sequencing, with the analysis tools that are available now, we are able to identify expansion diseases. And that is something that is also adding up to our yield. And it's also teaching us some interesting lessons. For instance, we had a family that came from, the father came from the northeast part of the country, and they were 3 brothers. Each of them married someone from the western part of the country. And then those 3 brothers, they died around the 50s with the exact same phenotype. And then the children started experiencing the phenotype at much earlier ages. So that mutation came from the northeastern part of the country, came to Kinshasa, and then so these 3 different ethnic groups got into those groups. We were able to find it was a spinocerebellar ataxia type 7. So we were able to actually to explain this to the family and actually let them understand where the disease was coming from and why those unrelated people would experience this just based on the fact that the fathers were from the same places, same family.
Fascinating and tragic at the same time, I guess, in this case. Do you have difficulty with, do people accept that there are genetic causes for these diseases, or is this a concept that is very difficult to explain to people, or maybe for various belief systems people just don't want to believe or just very reluctant to engage in?
I would say that we have different groups of people. We have people who accept easily. These are the care seekers. You have people that are really seeking for care at all costs. They would travel, they would follow to any hospital as long as they believe they can get the explanation. And then you have people that you have to convince, give some explaining to convince them that it's important that they go through the genetics. Sometimes I would say that this second group of people you have to explain is the best group as opposed to the first one, the care seekers. Because the care seeker, they will buy anything that you sell them, as long as they believe that they would get the explanation. This is the group where we have the bias consent, because they would accept anything you are putting on the table. We had that last week. We had a mother who came with a patient, a daughter with a developmental phenotype. And after the consultation, we wanted to have the talk go through the consent process. We have 8 pages consent form. We need to explain to them what sequencing is. We really use the language that is really easily understandable by everybody. Because the country is so religious, we use references from the Bible to explain to them what a genetic disease is, what sequencing is. So we need to walk them through this. And then the mother, that woman say, doctor, I think you are making it so difficult for me. I just came for the analysis. Why don't you collect samples from my child and do the testing? I don't need to go through all these papers. We stopped. We couldn't go further, and we ended the paper and say, okay, you go home, you read this, and then you come back when you are convinced that this is the way we should proceed. So the care seekers, at any cost, they would accept. This is the most dangerous group because they don't understand. They don't take time to understand what it is about. But the group where we need to do the explainings to convince them to come to us is the best group because they are more slow in engaging, but they are engaging with more understanding of what they are accepting.
Fascinating.
Yes, really fascinating. I am also so fascinated, Aimé, by how you are studying the facial measurements and the facial phenotype, because the DNA phenotyping is something I think very important in this morphology, and we don't use it a lot in genetics. We are trying to implement some kind of DNA phenotyping now in our patients to see how can we use this, for example, in patients with history of blah blah. Sometimes you have the parents, you might use their DNA in phenotyping the child and maybe try to draw a clinical picture of this patient or of their SIB to imagine or to have a provisional diagnosis of their lost child. So I'm very interested in how you mentioned you worked on the phenotype of the Down syndrome's differences between European and non-European patients and our African patients. Still, good luck with the challenges, because I can imagine how heterogeneous and how diverse everything is in Africa, not only in Congo. It's the main challenge in building any consortium any collaborative consortium in Africa. Whenever you start the discussion, okay, we had, for example, in the last time, what is the most important disorder, killing disorder, for example, in Africa? Yusru would answer with an answer, although we're both from North Africa, and you will give me an answer, and I will answer with the third one. So it's really so challenging. The nice thing is that you were speaking about training in clinical, which I think is a very important addition in your network, because it's always about bioinformatics and molecular data analysis. While in Africa, we actually miss the clinical diagnosis of the patients, how you wouldn't diagnose a genetic disorder at hundreds of clinics all over Africa, or even common disorders. For example, we couldn't deal with dengue fever patients when they arrived in Egypt from the Sudanese immigration. So please.
I think, Gada, you are raising a very interesting point. I see Yosri has the end. Yosri, would you allow me to
Sorry, okay. I thought you forgot your hand. Sorry, Yosri. Okay. I'm very sorry. So, please go ahead.
Yeah.
You have to speak up, Yosri.
Two things there, Gada. The first one is the facial recognition. That was a journey. It was really interesting. The first thing that we did is just test it, and it was developed by the company FDA developed that, and we submitted images of Down syndrome from Belgium and Down syndrome from DRC. The rate of diagnosis was very low in DRC. We set the threshold as, because when you submit a picture, you get matches. It's not clinical hypothesis because these are not clinicians. It's a tool. These are matches because it's trying to match 23 points on the face from the mask and to your patient. What we noticed that it was not efficient or accurate on patient of Congolese origin. So what we did, we trained the tool. We submitted 100 photographs of Down syndrome patient from DRC to train the tool. And at the same time to prevent the overfitting phenomenon. What we did, we provided images on non-Down syndrome, different ages. We fed the tool for that to improve the training. Then we were able to improve the recognition of the phenotype. Now, when we submit a Down syndrome, we are more confident that the tool is trained to recognize Down syndrome in African patient, at least African from this region of Africa where we collected photographs to submit. It was really a journey. It took time, about 2 years when we were engaging with the company to train this, because we wanted this to be something that people would be using. The second is about the training, the clinical training. We, as far as I can look, we have only like 2 or 3 countries in Africa where you have a clinical genetic training program. But the need is everywhere in Africa. Until we get to the point that we have programs everywhere, we need to capacitate people that are on the field working already, to really capacitate them, provide them with the genetic thinking so that they can incorporate this when they are seeing patients, and understanding of the genetic tests so that they know which tests that I have to prioritize for this phenotype. We actually have, in the frame of the DDD Africa, we have just started Monday. We started a course in Africa where we recruited duos, 1 clinician and 1 lab technician from 10 countries in Africa. And they are going to follow courses, 1 module per month until January. And in January, we will gather all of them in South Africa for a in-person training and the practical training in laboratory, because we believe that the skill is needed, but we cannot wait until we have trained geneticists. We need to train people that are seeing patients today. And we are developing a French component of the same course, because the course is in English. We are developing a French component of the same course starting in March, so that people will be able actually to participate. And
we suffer.
Yeah, the course is really the same course that people are following when you are training for genetics in Belgium, exact same course.
Great. Wishing you the best of luck in that. Yusri, please, sorry, I thought you forgot your hand. I'm very sorry.
No problem. Thank you. It's a very interesting discussion. And so as we are in the logical reasoning, I have 3 questions, but it will be very brief. So the first one is about how much whole genome sequencing can be useful in the clinical genetics or genetic diagnosis of monogenic diseases when compared to whole exome sequences. Mainly when we take into consideration the relatively high cost of whole genome sequencing compared to West, to whole exome. And when you are working in monogenic disease, you are supposed to sequence the trios. So does it worth it? I mean, what do you think about this point? This is my first question. The second one is about healthy data. And I know that you are involved in the Agenda project and different other projects, the Issue Africa. So what about healthy data and the importance of generating more and more healthy data from African population? And I mean, there are lots of questions about the number of healthy genomes that we need to sequence to get what we call the African pan-genome, because of diversity again in Africa and this ethnic diversity in the continent. And the third question, it's more, much more general, is about what motivated you to come back from Belgium to Kinshasa? So because we have young researchers here and they are really, really, I mean, many in Tunisia, we are really suffering from brain drain. And I can understand because of different reasons, but we want to hear from you. What really motivated you to come back to your country?
Yeah, thank you, Yusri. The first one, I think I'm not the right person to ask that question, whole genome sequencing and whole exome, because I'm a whole genome sequencing guy. really looked at the whole genome sequencing as, for me, the first tier for the analysis. Why? I would explain. In Europe, in USA, people would say, okay, why whole genome sequencing when I can explain about 80% of the diagnosis on exome? Because you did it. You did the exome and you did the genome. You know that if you do the exome, you have 80%, but nobody can say that about Africa. Nobody has done both whole exome and whole genome. We are not yet at the place where we can say accurately that the whole exome is better than whole genome in Africa. Second, the whole genome provides more possibilities. The same data you can reanalyze using the tools and identify, like I was mentioning, the expansions. You can explore the non-coding regions. At the moment, in terms of economics, I think the paradigm is shifting, because at the time it was one test after another. But if you have whole genome sequencing, just applying the genomic tools, you don't need to reanalyze. You are ultimately saving the cost over time, because you are now able to do the diagnosis today, and you can do the preventive medicine tomorrow. You can do pharmacogenomic study tomorrow from the same data. So overall, in terms of economic, that is my belief. I don't really think that is really cost saving at all. But we have the limitation. We don't have the money. We need to have project. That would be, for me, the acceptable limitation. But in terms of scientific place, we are not yet at the place where we can say for fact that the whole exome sequencing is what we need for Africa. We cannot, that's my belief. So the second was about, sorry.
Healthy data.
Yeah, the healthy data. We need more and more data from healthy individuals. We started that collaboration with ARDI like 4 years ago, but we failed to have people in Africa engage in generating those type of data. And I'll even go further. We not only need data from healthy people, we need data from healthy families to understand how some variant can segregate. If I have one single variant in a database, it's not sufficient. I need to know that these variants segregated in different modes, and it's not a variant that is manifesting because of the imprinting of the gene, because we don't know how, what is the full extent of the imprinting. Some genes are still to be understood. So unaffected families would be providing more interesting information in the future, especially in Africa. We need to have the understanding of the segregation of variant, not only the frequency of the variant, but the segregation of the variant is an important element. Third, why did I come back to Congo? People still think that I'm crazy because I took that decision. But it's more because as a clinician, I'm confronted with the misery of the families. I talk to families. I give tissue to mothers so that they can wipe their tears. And I see this, and I see how we practice medicine in Europe. It's more matter, it's like cosmetic that we are doing in Europe. People are coming to the hospital for things that I would say not really, that doesn't make sense. And you spend your time just listening to this. I know that people are dying to get the same service in the country. It's more about people. I always say to people that I work for the patients. My boss is the patient. My boss is not the director of the hospital. I'm one of the most difficult people to work with because I pay more respect to the patient than to the regulation of the hospital. This is really the driver that brought me back to the country. And on top of that, if people didn't have opportunity to read my World View in Nature last year, you can read my World View in Nature to understand my history, where I'm coming from. I was so sick when I was a baby that my parents promised, they promised that if I was healed, I would become a pediatrician so that I will be able to treat patients myself as well. So it's really a long history that is rooted in me since I was a young boy. And yeah, I couldn't escape that. It was my fate. And I think the best place to do that is the place where the need is the biggest, and the need is the biggest in Africa. You can do that in Africa and get money. You can do that in Africa and get recognition. I'm today sitting here. We didn't know each other with Joseph, Harold, and Joe. So it can bring you everywhere. If you are doing your science in Africa, you are not forgotten, but you have the opportunity also to be recognized in the scientific field. And you also have the opportunity to live a decent life if you are good in what you are doing. So there's no need to put yourself under so many stress of being a foreigner in another country. It's too complicated.
I would also, yeah, I was just gonna say, I would add that I always tell people, like, when you are in the developing world, you have the actual more important resource than we have in the West for genetics. You have your populations. They're understudied. There's so many interesting questions. So that's a huge advantage you have being there that no one in Europe has. So there's so many reasons that what you're doing is the right thing. You know, it's great.
It's the best thing to end our time with. Actually, regretfully, we came to the end of our allowed time. Thank you so much, Aimé. I really appreciate your presentation, your participation. Thank you all for a very nice discussion. I hope that your photo of NASA, sorry for the noise, it's from the street. Someone is having his car, maybe for a background music to end the thing. So,
thank you for the invitation. Yes, I really enjoyed being here.
Yes, we enjoyed your presentation. We enjoyed the discussion. I hope that your NASA photo the next time would have more light in it. Thank you. Thank you so much. Bye.
Thank you. Thank you, Aimé. Thank you all. Bye-bye.
Bye-bye.